No way.
Hi, can you hear me now? Oh my goodness. I'm so sorry, buddy. This is like almost the end already. Okay, hi, I am Mr. Jason Pang. I think for 10 years already. Please join us. Okay. Right, so okay, sorry about that. I mean, then the recording is also slow. Okay, so apologies for that. Okay, so okay, this is just my face. Okay, okay, see me there. All right, what else can I show you all? Okay, so we just want to work on the topic for today. We have 4 things to look at, right? So in 1.5 hours, we're not going to be able to go into detail for all of this. As the name suggests, this event is a crash course. We're gonna hit it hard and fast and make sure we can get through as much as possible. So I will depend more on your questions. You don't know, you tell me, I tell you, then you know. Okay, 2-step process only. Okay, so ask questions, feel free to work on it together. And most importantly, I feel that it's the last point. You look at the pink little bird over there, according to your school schedule. right? So if you do get to a class going, tell me what is your schedule. And it's very simple because all of you, one schedule only, same class, same school. Can? Okay. So this one will be more for when we start a class. Now I'll get to know you all a little bit more. Okay. You'll recognize this code. Am I too old? I mean, I'm not born in 64, please. Okay, it's just the show was in 64. Okay. Okay, but without further ado, let's head on to our friends online. Make sure that we're looking at excretion first. Okay, we're looking on excretion first. Oh, wait, excretion. Okay, let's go. Right, so I heard some of you all have been working on it already. Okay, so fairly straightforward. We have 4 topics, 4 worksheets. We're gonna hit it hard and fast, as I said earlier on. So look at the questions. If you look at the questions already, I will not just ask for answers. You want to be able to make sure you can revise properly as well. Okay, right? So why is glucose found in the urine of diabetics? Right. So here we are thinking of, is it naturally found in the urine of a healthy person? Would glucose be found naturally? No, absolutely not. So why would it not be? We must first think of glucose being a nutrient, something that good that we want to keep in our body, right? But as with everything, as the saying goes, right, too much of a good thing, it's going to be bad as well. So that would be diabetes here. In this topic, excretion, what is the organ we are looking at? In the topic of excretion. Tell me the answer, please. In the topic of excretion, what organ are we looking at? The kidneys, thank you. Okay, so in this case, glucose in the urine of a diabetic, is there anything wrong with the kidney? Is there anything wrong with the kidney? Glucose in urine of a diabetic, there is nothing wrong with the kidney. Okay, so this is a common error. There's nothing wrong with the kidney. Okay. Please follow along. Okay, so please keep this in mind. Right, if glucose is found in the urine of a diabetic, there is nothing wrong with the kidneys. So you must always understand. So if let's say in a healthy person, if there are 10 units of glucose in the bloodstream, all 10 will be removed via what process? By what process? There are 2 processes that occur in the kidneys. The one that removes stuff first. Excretion is the entire idea. Ultrafiltration first, ladies. Remember ultrafiltration first. So all 10 units of glucose will be removed via ultrafiltration. But in a healthy person, all 10 units will later on be selectively reabsorbed. Can? Okay. So in a diabetic, I'm remembering some names. Lini. Hi. Okay, you choose first. Okay, I asked. Alicia. What's my question? Oh, what does it mean to be a diabetic? What's the symptom of diabetic? Abnormally? And normally high blood glucose. So just now the healthy level was 10. Now I'm going to say this person is 50. Okay, so all 50 will still be removed via ultrafiltration, but only still 10 will be reabsorbed. Where does the remaining 40 go to? If all 50 were ultrafiltrated, only 10 will selectively absorb. The remaining 40 will be excreted in urine. So that's the idea here, right? In this case, if this symptom is achieved, right, this person may have a functional kidney, healthy kidney, still reabsorbing 10, but it's just that there's just too much glucose already. Right? So must always keep this in mind. In bio, usually we always say like one thing wrong only. If this question say this person is diabetic, don't give this person kidney failure as well. Okay, so always keep it to one thing, keep it simple. Okay, okay, right. So over here, why is glucose found in urine of a diabetic? We are looking at D. Not enough glucose is converted to glycogen. Right? Okay, so what we're looking at here is option D would mean that the kidneys are not functioning. Correct. So that as I just explained, we don't give this person multiple diseases. All right, let's be a little bit more kind. Okay, so not enough glucose in the blood is converted to glycogen. That's why abnormally high blood glucose, like what Alicia helped us with. Can? Okay. Okay, moving on. We're looking at reabsorption of water, primarily occur in the kidney in humans. So can we underline or highlight the word primarily? So would the question change drastically without the word primarily? Let's think about it. All right, now will the question change drastically without the word primarily? All right, I need us to remember. Even in the tubules, water is reabsorbed. In the what are the 3 tubules? Alyssa? Very good. And finally, LOH. Wonderful. Loop of Henle. So in all three tubules plus the collecting duct, water is reabsorbed. Water is reabsorbed. Okay, so here we're looking at the primary means of reabsorbing water. So answer should be the collecting duct. Okay, so please take note, because if I don't have the word primarily, all four answers are correct. Can? Awesome. Let's go, let's go. Here we're looking at, okay, moving on to dialysis now, moving on to dialysis. Simply put, this one, then got kidney failure lah. All right, this one got kidney failure. We need to help this person filter the blood. We need to help this person filter the blood. We're looking for the colors and the shapes here represents urea. So blood in will have a lot urea. Blood out will have very little urea. So this one, fairly straightforward. Look at your colors and shapes only. Lenny, back to you. Come, your answer question 3 should be the triangle. Simple as that. Can? Okay, so there should not be any triangles returning to the person's bloodstream. Okay, so theoretically, this is similar to some question Lenny asked me last time as well. So in our syllabus, we just keep it simple. There's no urea return. But realistically, there may be some, right? We cannot achieve 100%. Can? Right? Realistically, we may not be able to achieve 100%. Okay, awesome. Let's go. Flow rate and concentration of protein in urea. Okay, blood vessel leading into the kidney. Can we have the name of the blood vessel? Blood vessel bringing blood to the kidney. Renal artery, very good. So renal associated to the kidney. Artery means away from the heart. Oh, away from the heart means going somewhere, going to the organ lah. Can? Okay. Oh, can I not just send? Who's this? So 3, no, not yet. Oh. This person. Yes. Okay. All right. So what are the correct figures for the fluid in the collecting duct? Okay. So firstly, proteins. Should I have proteins being removed via ultrafiltration? Absolutely not. What ensures that proteins do not get removed via ultrafiltration? Jason, once more. Basement membrane. Okay, the basement membrane, think of it as just like a net wrapping around the glomerular capillaries. Okay, keep that in mind. So pores of the basement membrane wraps around the glomerular capillaries. So only those small stuff can get through. Okay, but keeping in mind, small stuff, there's no indication whether it's good stuff or the bad stuff. As long as you're small enough, you will get removed from the bloodstream. Right? So there's no indication of good and bad. So therefore, our answer over here, definitely we should be looking for A and C. All right, not agree? A and C, we should not have any proteins. Protein flow in the collecting duct already. All right, now collecting duct, that's kind of like pre-urine already. Right? Fluid in the collecting duct is kind of called pre-urine already. So no proteins. So between A and C, we are looking at urea concentration still the same. So flow rate. How? Flow rate, how? Is it going to be 10 or is it going to be 1,000? So they gave us the information. Flow rate in the glomerulus is 1,000. Heading to the glomerulus is 1,000. So should it increase or should it decrease? So should it be the same or should it be smaller? Mehan? Was it Mehan? Come, your explanation. Which one do you choose again? You chose A because? Then? Why would the flow rate in the collecting duct be lower? Okay, so efferent, arterial, wider lumen, and efferent, right? So I'm creating a high what? What's the last word you say? Pressure, right? Then this question, where is it? This one is about what? This one is flow rate. It's not pressure. Right now, so immediately you look at the quantity I'm comparing is different already. Right? So you are absolutely correct to say that there's a higher pressure at the glomerulus thing. But here we're looking at flow rate. So how much flows per time? versus how much pressure the fluid is exerting on the vessel walls. Okay, so therefore, if 1,000 enters, 1,000 will flow as well. Okay, just that the 1,000 in the glomerulus experiences a higher pressure. Can? Okay, flow rate, so it's the amount of fluid that flows per time. It's different from pressure, right? The force exerted by the fluid on the vessel wall. Okay, slightly different. Okay, so again, we are looking at the stuff that can flow in and out from various parts of the kidney. Now, it's very important for us to state mammalian kidney. Our topic here is focusing only on the human kidney. So focusing on the mammalian kidneys. Different organisms have different adaptations to their kidneys. Some have longer medulla, some have smaller medulla, right? Okay, so that's where the renal pyramids are. Okay, so over here we are looking at how all these substances change in concentrations. Okay, how these substances change in concentrations, right? So X, Y, and Z we have. Okay, on the first one we have X dropping down very quickly, and there's none in the Bowman's capsule. There's none in the Bowman's capsule, none in the tubules, none in the collecting duct. What would that be? What would that be? Okay, so X you chose to be what? X to be protein, right? So this is similar to just now what we just said about Bowman's capsule, right? Okay, excellent. So X only a protein, we're looking at C and D only. Okay. Okay, looking at C and D only, X will be protein, right? So between urea and glucose, you know, you will choose D. Okay, can you explain why there will be higher glucose than urea at the start? So in the glomerulus. Why are the concentrations the same in the glomerulus and the Bowman's capsule? I think it's reabsorbed. Yeah, very good, right? So everything that can be removed by ultrafiltration from the glomerulus will enter the Bowman's capsule. That's why it should remain the same. Okay, so because we have reabsorption, glucose will go down and urea remains there. Okay, but if you look at this, this one, Z is urea. Why does the concentration go up? Do I have more urea? Does the amount of urea increase? No. So that's why you must understand again the meaning of concentration. The amount of urea is the same compared to the fluid, the solvent decreases, so it becomes more concentrated. Right? So this idea here you must keep in mind amount versus concentration. Right? It's a very, very commonly overlooked point, amount versus concentration. Okay, amount versus concentration. Okay. Okay, so as I shared earlier on, we have a lot of things to go through, right? So I'll pass you all the answer key after the lesser one, right? So we're just going to skip through some questions. The reason why I have put more questions than we can possibly cover today is so that you all can have an opportunity to practice other questions as well. Okay, right? So we're just going to go through very quickly first. Definition for excretion and why it's important. The removal of what? For myself, I always remember TMW, toxic metabolic waste. Okay, so toxic means that if I don't remove it, it will cause harm. Okay, if I don't remove it, it will cause harm. So that's toxic. What does metabolic refer to then? Toxic metabolic waste. The metabolic refers to what? What would a metabolic waste be? Produced. Very good. A metabolic waste is something that we produce versus feces. We did not produce it. It's something that we consumed, then we never break down, so we remove it. So that is one of the core differences between excretion and egestion. Okay, that's the core differences between excretion and egestion. We remove stuff that we made, so that's excretion. We remove stuff that we could not do anything about, that's egestion. Right? Okay, so let's keep that in mind. Explain its importance is related to the metabolic thing and the toxic thing. If we keep it in, it will cause harm. Okay, if we keep it in, it will cause harm. Other than urea, what are some other things that we need to remove via excretion? Okay, so all the nitrogenous waste, urea, uric acid, creatinine, these are the nitrogenous waste that we remove. What other things do we need to remove via excretion? Are there other things that we need to remove via excretion? What else? A bit of water, so excess water, right? Hey, 11 minds here. Come, share with me, please. Other than the nitrogenous waste, other than excess water, what else do I need to remove? I'm removing it right now. Yes, carbon dioxide. Carbon dioxide, if it accumulates, what harm can it cause? You know, blood pH. Carbon dioxide is an acidic oxide. If it accumulates in the bloodstream, blood becomes too acidic. Why is that a bad thing? Other than the fact that acid is bad, right? But specifically, if they ask you for an example, why is it? What is the harm that comes about when blood becomes too acidic? What does pH affect? Enzymes and proteins. Those are the nature, isn't it? Okay, so keep that in mind. All right, thank you, Jason. Okay. So simply put, recall questions like what is where, all these things need to have at your fingertips. So ADH, where does ADH primarily function on? Which structures? Collecting duct, right? So we should be able to identify the collecting duct as this one, this one is my collecting duct. Okay, this structure is the collecting duct, very straightforward. Okay. Let's jump ahead. Your comparison, what would be your guess be for C part 1? How much glucose would there be at in region W? Let's first identify region W. Sorry, what's your friend's name? Zhe I remember Zhe. Okay, sorry, Zhe W is what? Oh, the previous page is diagram. W. W distal or proximal? Why is it the distal? Okay, so first and foremost, W is neither collecting ducts. What is W? It's the loop of Henle. Hey, let's see properly. This is the entire loop. Can you see that? The convoluted tubule is very obviously drawn. It's going to be a mess like that one. It's going to be like that one. Can you see that? All right, so please take note of that. So PCT, loop of Henle, DCT. Clear? And all right, PCT, loop of Henle, DCT. So we have W as a loop of Henle. And actually, it's the one that's almost reaching the distal already. Okay, so at this point in time, as the filtrate flows in this direction towards the DCT, most of the selective reabsorption would be almost complete already. Okay, loop of Henle. So moving up to the DCT, most of it should be complete already, almost complete. So here, we should be looking at 0. Why? We should be looking at 0. So simply put here, it's just reabsorb. For 2 marks, why would that be 2 marks? This question is asking us to complete the table. And what does the table do? The table compares the 2 regions. I understand what the question is looking for. So question asking us to look at the table. What does the table present? The table presents comparisons between these 2 regions, X and W. So if we are asked to complete this value, our explanation here for 2 marks is why here have, why here don't have. So why in region X we have glucose? What is region X? And therefore, why is there glucose there? Bowman's capsule, right? So why would there be glucose in the filtrate in the Bowman's capsule? That's ultrafiltration, right? Okay, ultrafiltration only allows the small stuff to pass through, and glucose is the small stuff. Okay, so let's keep these points in mind. Let's keep these points in mind. Right? So our first mark is why X have. Then our second mark is why W don't have. Very straightforward. Selectively reabsorbed. Okay, all of it would have been selectively reabsorbed. The PCT and the loop of Henle. Okay, and right. So similar idea. If you can understand what we did in part 2. Part 3 here is asking us to compare urea concentrations. Okay. This is actually something I kind of explained earlier on. So you think about urea concentration. The table shows us that there's an increase in the concentration. Actually, can you help me with this, please? The urea concentration increase. Does this mean that I put more urea into the tubules? Very good. Thank you. Water is selectively reabsorbed at the convoluted tubules. So I have less solvent, but amount of urea remains the same. Hence, urea concentration increases. Following? Yeah, very straightforward. All right, awesome. Finally, moving on to ADH. Where is ADH produced? Yeah, let you finish first. Okay, now don't need to stress too much about writing all these things. I'll pass you the answer later on. So just point form that works for you. Okay. As you're writing, help me think of the answer to where ADH is produced, please. What organ produces ADH? Yeah, where is ADH produced? Produced. Okay, released by pituitary, yes. Hypothalamus is the one that produces it. Hypothalamus produces ADH, stores it in the pituitary. The pituitary is the one that regulates the release. You all definitely got learn hypothalamus, right? Got learn hypothalamus? Oh, no, as in just like overall, hypothalamus got learn this word. That's right. Oh, by the end of the day, like the end of yours, I think. That's yeah, right. Okay, Oh, osmoregulation not tested. Okay. Osmoregulation not tested in the upcoming WA lah. Is it? Not tested. Okay. No worries. Okay, then we will skip the ADH question. Okay, ADH is the one that is involved in osmoregulation. Okay. Yes. Learned it, but not in the WA lah. So yeah, and is it? Alright, is it? Do we agree? Is it? Richard say not. Lini also say not tested, right? Hey, go and check. Can all right. Okay. So I would like to actually move on to the next topic already. Any outstanding questions that y'all want to clarify first? Can? Okay. All right. So we'll move on to looking at cell division now. Let's go in cell division. Okay, can we do 1, 2, 3, 4, 5. Okay. Yeah. Let me go and take it. Cell division. Cell division. Okay, I said. Okay, here you go. Does everybody have cell division? Everybody have cell division? Okay, just gonna put it here. Okay, so who Yes, I'm gonna send it to her now. Is it? Some. Okay, cell division. Okay, cell division. What is one of the biggest challenges that you are facing for cell division? Are you all like okay with cell division? Because if I'm honest, cell division can get confusing. You all tested on everything, mitosis, meiosis, everything. Okay. When does the chromosome number get halved in meiosis? Is it after meiosis 1? Do you agree? When does the chromosome number get halved during meiosis? After meiosis 1. Do you agree? Okay. Okay, very good, correct. Sorry, what's your name again? Legra, yes. Okay. Sorry about that. Okay. So what I always like to one of the biggest things that we're thinking of, right, is at the start. This is one chromosome. All right. What is this process that results in this replication? How many chromosomes do I still have? Still one, okay. So let's say I'm a diploid organism. So I have 2 pieces. Therefore, again, 2 pieces, okay, 2 chromosomes, diploid, right? So if I'm looking at meiosis, my first division will already result in me having, okay, this is one cell. I'll have. Therefore, now at meiosis 1 is already half already. Previously is 1 cell, 2 chromosomes. Now 1 cell, 1 chromosome already. Can, right? So in meiosis 2, I will now have on this one will split from the other side. So therefore, I now have, and the same for the bottom. Okay, so one of the biggest confusions, right, that over the years that we can see, right, is this not understanding at which point does the reduction occur, right? So keep that in mind, wonderful, Legra, thank you. Okay, so that's just one of the first few things that came to mind that I wanted to make sure we are all on board with. Okay, so order of events, I'm sure we can all remember PMAT. Very straightforward, PMAT. Okay, so what happens before P? What's that thing called? Interphase. Actually, we can split them into 3 things, but you only need to remember the middle one. What's it called? DNA replication occurs in this. What's the name of this phase? S phase. S for synthesis phase. Okay. The first and third gap phases, they just growth. You can think of it as just growth phases. The cell gets larger, the cell makes organelles, so that later on you can prepare for cell division. Very straightforward. Okay. So just remember DNA replication in S phase, and then you have PMAT, followed by one more. After T, you have cytokinesis, right? Cyto of the cell, kinesis moving apart, right? So kinetic thing of it moving, right? So cytokinesis is the actual splitting, okay? So if the cell is arrested after telophase and doesn't get into cytokinesis, what is the state of the cell now? You visualize the diagram that we should be looking at. If the cell is arrested right after telophase, doesn't proceed into cytokinesis, what do I have now? 2 nuclei. Very good. 2 nuclei, and the cell is just one elongated oval. Okay, right? So at this point in time, this one cell has still the same number of chromosomes as before. Okay. All right. So if you're looking at PMATC, fairly straightforward question over here. Alicia, question 1, you will choose. Sorry? I can't hear you. C or D? You say C or D? C. So we are beginning with number 4, chromosomes uncoil. What does it mean to uncoil? Okay, right. To uncoil means is to decondense. That's not what we're looking for. We should be starting with number 5. The chromatids are joined by a centromere. When does that occur? Replication. Right. So we start off with 5, right? Joined by a centromere, and then you're gonna line up along the equator. That's our metaphase. Then we have being split. So that's anaphase. Followed by moving on. So that's to opposite poles already. And then we have our uncoiling. So that's in telophase. Okay, first one answer is 5. Answer is D. Okay. Mitosis, 1 division. Oh, thank you. 1 division of mitosis. I start off with that cell. Mitosis gives me how many daughter cells per division? 2 or 4? 2. Very good. Okay, mitosis, we're looking for genetically identical daughters. So therefore, I should be looking for the one that gives me literally identical pictures. Okay, literally identical pictures. Zixing, you chose. Sorry? B, wonderful, B, thank you. Human cell, how many chromatids and is the nuclear envelope present before prophase? Before prophase. Alyssa? Okay, 46 chromosomes, right? So this one chromatid, is there a difference? This is one chromosome. This is one chromatid. How many chromatids are there? There should be 92. Do you see that? Can you see the difference? Okay, Mehan, clear or not? So this is one chromatid, then this is one chromosome, while this is one chromatid. Then this is another chromatid. Like that. So I have two chromatids per chromosome. Make sense? Okay, so you should be looking at 92, and it's a nuclear envelope present. It's a nuclear envelope present, definitely. So only in prophase, then you will break down the nuclear envelope. Okay? Before, yes, before. Okay? Okay, so this one fairly straightforward. This one is just a numbers thing. If mitosis is genetically identical versus meiosis, we have reduction division. Question 4, we should arrive at C. Okay? All right, start out as 8, so I will still have 8, then afterwards I half it. Okay, here we're looking at gamete. So we should think of meiosis already. Gamete, we should be thinking of meiosis already. What are two very unique events? Lini, can you help me with this, please? What are two unique events in meiosis that do not occur in mitosis? Wonderful. Crossing over and independent assortment. Mehan, crossing over occurs in prophase 1. Very good. Richard, independent assortment occurs in metaphase. So it can occur in both metaphase 1 and 2. Because in metaphase 1 is like that or like that, versus metaphase 2 is like that or like that. You know what I'm doing? Like that, right? You understand? Okay, very good. Okay, so here we should be looking out for crossing over, right? So we should not have a nice shape over there. Okay, so we should be looking at interspersed. So if we do not want to look for solid lines, A is out. A is mitosis already. Allegra, you chose? C for correct answer. What happened to D? What happened to result in D? Yes, Mehan, what's that called? Non-disjunction. Remember that? Okay, so failure to separate properly during anaphase is known as non-disjunction. Okay, so I will result in a gamete that is, do you all know the name for this condition? Aneuploid. Have you heard of this word before? Aneuploidy is when you have a wrong number of chromosomes, right? So non-disjunction, come, I spell it on screen. Oops, too fat. Non-disjunction will result in aneuploid cells. Okay, so to be aneuploid means to have an abnormal number. It doesn't mean a fixed error. If you have more or less, as long as you're not the correct number, you are an aneuploid cell. Simple as that. Can? What is your syllabus only got one consequence that you need to learn? Down syndrome. What's another name for Down syndrome? Trisomy 21. Try me 3 copies. What does somy refer to? What does the somy refer to? Trisomy. Somy here, we think of autosome. Can you tell me what's an autosome? Thank you. Non-sex chromosome. So 3 copies of a non-sex chromosome. That is trisomy. Which non-sex chromosome is it? 21st. That's why trisomy 21. Can? Okay. Is this the only type of chromosome mutation that occurs? Is trisomy 21 the only kind of mutation that occurs? No, it's just the most common. And I want you to think about it. If it's the most common, it is the least damaging. Does it make sense? It's the most common because it's least damaging, because this person can still survive. other types of mutations, maybe trisomy 14, the zygote, the embryo cannot even undergo proper cell division. So it will have its self-termination. So that pregnancy will be terminated just there. Okay? All right. So yes, answer is C. Very good, Allegra. Okay, so here we already say that we have crossing over. Okay, but what is this one event that must happen before for crossing over to occur? They must pair up to form a bivalent. The pairing up is known as, sounds similar to a term in nervous system. Synapses describes the pairing up of a synapse. Is the gap between neurons. Okay. 2 points here. We need to state what makes chromosomes homologous. We have 4 things to look at. What makes chromosomes homologous? What makes chromosomes homologous? Same what? Size, shape, and length. Very good. Same size, shape, and length of the chromosomes. Then our 4th one is same size, shape, and length of the chromosomes. Okay. Yes. So this, therefore, okay. For example, if I use letters ABCD, ABCD, same size, shape, and length. But if this one is ABCD, this one is ACBD, not homologous anymore. What am I looking at? They must also have the same one. What's the name of this? We don't use gene, we don't use position, we say loci. Okay, gene loci. So therefore, we are looking for gene loci as well. Okay, loci is plural, locus is singular. Okay, so same total length and number of genes. Okay, yeah, loci. So the points of crossing over, P and Q, what do we call them? Chiasma, if you write down chiasma over here, no marks. Why? Very good. Points P and Q, chiasmata. That's like stoma, stomata. Plural will have to be chiasmata. Okay, please keep that in mind. Importance of crossing over. Importance of crossing over. Alicia, can I hear from you, please? Why? What benefit? What advantage does crossing over give us? Increases something. Alicia, what do you increase? You know? Jason, help me, please. Genetic variation when forming what? That's very good. State the gametes. Because if I'm not forming gametes, I would not want variation, isn't it? Okay, right? Genetic variation when producing gametes. 2 places in plants where meiosis takes place. Places in plants where meiosis takes place. So don't always just think animal cell only. Risha, 2 places in plants where meiosis takes place. Never mind. Then think back when you were P5. Repro implants. Sorry? Enter, correct. Not stigma. Stigma is just where the pollen grain land. Yeah lah, ovary, ovule lah. As long as you have gametes inside, then it will occur already. Can? Okay? So that's why even if you haven't learned repro yet, doesn't matter. It's a simple enough question. Okay? Enter. ANTHER. Okay. Yes. The filament, enter. The male, yes, the enter is the one on top. Think back to P5. Okay, very common question as well, giving you actual photomicrographs. So this one, the scientists look at the cell, say, hey, divide, then take picture, then you put like that. Looking at the pictures, you must be able to identify what is going on. First question, very important. Lini, can you help us to identify what type of cell division is this? Meiosis. Why meiosis? Which diagram are you looking at? And the information you're looking at in B is for what? Very good. 4 daughter cells. Okay. For my entire life, every time Somebody asked me why I always use the girl and why daughter cell, why everything sister chromatid. Oh, I don't know. It's just what they use. Okay. All right. So for daughter cells, very good. So it's meiosis. And same type of question in A part 2. What part of the flower would the scientists have looked at to obtain these pictures? Yeah, all those are the same answer as just now. As long as it's an organ that produces gametes, you will have this occurring already. Can? Okay. Now identifying the stages. What's happening at A? What's happening at A? We have 2 daughter cells only. Correct or not? We have 2 daughter cells, but they all realize that the pooling is occurring in this direction. Can see that? So it has to be A, then afterwards it splits, then B. These are the last 2. Make sense? These are the last 2. So 5 and 6, A and B respectively. Okay? Both are meiosis 2 already. This is 2 daughter cells that after they split will form this. Can you see that? Yeah, this one is anaphase. Then you have one cytokinesis, telophase here, then cytokinesis to form here, Clear? Clear? Okay. So what is happening at C? Let's look at what is happening at C. Anything? Any words you can use to describe what's happening at C? They line up along metaphase plate. Kind of, right? This way. You see that? All right. Okay. So more obviously, we can see definitely we will know what is happening at D, right? What is happening at D? They are put away, right? A for anaphase, A for away. So if this one is metaphase, this one is anaphase, right? So therefore we have C and D in this order as well. So keep this pairing in mind. All right, C and D. Then we also have A and B. Okay. Then we have E and F. Oh, my goodness, what's happening at E? Why there are like chunks all over the place? So what are those black chunks? The genetic material, the chromosomes, right? So if they're scattered all over, what stage am I at? Prophase. Very good. Because they have not been aligned. Okay. And in F, what is happening at F? What's happening at F? Yes, the thing. But telophase what? You are correct. It's telophase. Telophase 1. F is telophase 1, where you have the daughter nuclei put to opposite poles. So only after this, then you go through prophase 2, metaphase 2, then anaphase 2. So with that, what's the order? What's the order? Richard, read out to me, please. Wonderful. E, C, D, F, A, B. So look at picture E to find the haploid number. Picture E here. Literally, count the kids. You're printing clear enough. No? Ten. How to count 10? Okay, come, ladies. Ladies, on screen, yeah, count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So is the answer 12? It is 6. We're looking for haploid, right? Looking for haploid. Okay, so over here, our representation will be 2n equals 12 and equals to 6. Okay, keep that in mind. Okay. Okay, let's keep on ahead. I want to look at part 2. Why is meiosis important in reproduction? The first answer is what I asked Alicia just now. Alicia, what is the importance in meiosis? I asked you something just now, right? You remember? What's the answer? When producing gametes. Very good. So why is meiosis important in reproduction? I need to ensure that my gametes have genetic variation. Okay. What is another importance? Which I would say is actually even more important than genetic variation. by producing diploid first. You start that, then say I can restore diploid. Right? So yes, very good. So one of the very big importance of meiosis is to produce haploid gametes, so that upon fertilization, I restore diploid number. So you switch off the produce haploid first. Okay, right? Because I produce haploid, that's why I restore diploid, because n plus n equals to n. Okay, n plus n equals to n. Okay. Okay, so very briefly, running you through those. Yeah, jumping ahead. Again, jumping ahead. Fairly straightforward. Okay, but I want to ask first, what is this process? Figure 2.2 shows me what stage of division? Anaphase. Okay, and then afterwards, what happens? Anaphase. Then afterwards, what happens to an animal cell? Telophase, then cytokinesis, right? So anaphase, telophase, cytokinesis. Part 3, the purpose of me putting this question is, how would these processes from anaphase onwards differ? How would it differ in a plant cell? What do I call that thing in the middle? You all say it just now, I heard the answer. What is it? Cell plate. All right, cell plate. Right, you must understand, in animal cells, how cytokinesis works is literally, you look at my hands like that, it will just pinch inwards. What do I call this action? What forms? A cleavage furrow. Okay, so actually how this works, right? Just imagine I have a ring of contractile proteins. These proteins will literally squeeze inwards. So imagine yourself wearing a belt, then your body like that. That's it. That's what happens in cytokinesis. Okay, plant cells cannot have that because they have the very rigid cell wall. Okay, so instead of cutting themselves up, they form another cell wall to separate the two daughter cells. Simple as that. Okay. Right. So partly the idea here is we want to compare cytokinesis in plant versus animals. Okay. So cytokinesis in animal cells, we have the cleavage furrow forming, right, that pinches the membrane inwards to separate the daughter cells. Okay. Cytokinesis in animal cells is as such, right? The cleavage furrow forms, pinch the membrane inwards to separate daughter cells. Okay. In plant cells, you have cell plate in the middle, the cell plate coming in between the two daughter nuclei, and then forming a new cell wall. Okay, so moving on to this one. If I tell you that I write down the answer for 4A as having 46 chromosomes, 23 from father, 23 from mother, would that be a correct answer? It is for 4A, if I say to have 46 chromosomes, 23 from the father, 23 from the mother. Mihan says wrong. Why? Very good. Okay, keep that in mind. The number is only specific to humans. So to say your answer should be very good. 2 sets of chromosomes. Okay, one paternal, one maternal set. Okay, okay, awesome. Right, keep that in mind. Okay, is this sexual or asexual repro? Lini, can you help me with this, please? Is this sexual or asexual repro? Why sexual repro? And therefore, what process occurs? Very good. Okay, so one of the key defining properties of sexual or asexual is whether or not you have fertilization of gametes. This has a bigger emphasis than how many parents. This one only got one parent, correct? Only got one parent. Okay, so keep that in mind. If there are gametes and they fertilize, then sexual already. But why would there be little genetic variation compared to a two-parent sexually reproducing process? Why this has less genetic variation than a two-parent sexual reproduction? Natural number is lesser, what do you mean?
The 2n.
The 2n is always going to be 12.
Then when is 2?
Oh no, okay, let's say I have this organism is 12, 2n equals 12. This one also 2n equals 12. But this one undergoes this way. The other one has two parents.
One parent, so the same DNA.
Very good. This way, I only have one pool of genetic material to split. I only have two ways to increase variation. I have crossing over, I have independent assortment. Whereas in a two-parent situation, I have two different sets of genetic material to play with. Okay? This is actually, you learned before one, cross and self-pollination. You ring a bell? In self-pollination, same parent, but sexual repro. In cross-pollination, different parents, no genetic variation. Okay? All right, awesome. Okay, this one is a little bit on molecular genetics. Have you all done molecular genetics yet? Genetic engineering also do it. You extract the gene of interest, restriction enzyme, and then your DNA ligase, all this. Okay, so this is a simplified diagram showing what would happen over here. We are okay. So what is the division at P and Q? Frog A, P, egg. Ovum, you know, P is process that produces the gamete. Only two mitosis, meiosis. Which one produces gametes? You know. Meiosis. Very good. Okay. All right. So Q for egg develops into tadpole. So this one is all of us and that's mitosis. Okay. From the zygote, you will develop into the embryo and ultimately us. Okay. In this case, the baby frog, which is like a small little chunk of green, who is it a clone of? A or B? B, frog B, right? Because the genetic material is from B. Okay, genetic material is from frog B. Okay. Okay. Yeah. Okay. That's it. So basically, see, this is one of the most basic questions that we can think of. Or what the chromosomes do, they start to condense. That's prophase. Move to the metaphase plate. Metaphase. Pull apart. Anaphase. Decondense. Telophase. That's it. Okay. So one word we assigned to each phase of cell division. Okay. Okay. So questions from excretion and cell division. Anything? Yes, right. So just keep thinking of that. Okay, so PMAT. P for prepare, M for middle, A for away, T for opposite of P. Okay, let's keep that in mind. All right, so questions for these two topics. Okay, let's move on to the next one. Shall we do respiration or transport first? Wow, outpouring of transport. It's one of my favorite topics to look at. Okay. Yes, okay, we have 5 over here. Here you go. Transport. Up here you go, transport. Okay. Oh, sorry. Oh, okay, only so far so. How many copies do we still need? Who do I have transport? Do you have transport? No, do you have transport? I'm missing 4 copies. Okay. Okay, here you go. Wait. Sister Lee. Sister Lee. It's also not diastole, diastole. Go, here you go. Who is the systole person? Oh, have it. Oh, you have. All right. You need a stapler? Oh, you have slipper. Slipper. Anybody slipper? Okay, come. All right, so let's go. Friends at home, transport in plants, please. Transport in plants. No, what plants? Humans. You need. Okay, let's have a look. What is systole? Is it contraction or relaxation? Contract. Systole is contract. So diastole, I want us to think of diastole as dilate, becoming larger, relax. Right, so if that helps, diastole, dilate, larger, relax. Okay, so systole is the opposite. Okay, identify X and Y, please. What are X and Y? Which ventricles are they? Who is left? How do I identify Y to be the left ventricle? Okay, thicker left ventricular wall. So that's why it's left ventricle. So when the ventricles are contracting, which valves open, which one closed? Simple as that. Okay. So firstly, this one, can I just have everybody work with this on me? So let's choose, maybe let's look at valve 4. What is valve 4 called? Okay, bicuspid. So now ladies, work on this with me, please. This is something that I think will help a lot. Bicuspid. Okay, Richard, you want to talk about open or close? Open or close? Okay, open. So in what instance will the bicuspid valve open? So if the bicuspid open, first thing you must identify what is it between. So bicuspid is number 4. So I have left atrium flowing into left ventricle, correct? So over here, I'm just going to do it this way. Very straightforward. LA, LV. Okay, there's a reason why I'm drawing it in this order because I can use that as an arrow sign, yes, right? Blood flowing in this direction. I can also use it as an inequality sign. We call it. If you can come to this point, all other valves are the same. So if you look at this one, open means blood flowing in the correct direction. LA, flow correct direction in LV. Then why does this occur? Pressure is greater in than the LV. So if pressure is greater, means this part. Make sense? You can get this, all the valves are the same. All the valves are the same. So if I look at, aortic valve closed. Aortic valve closed means blood flowing correct or wrong direction. Aortic valve closed. Blood wants to flow in the wrong direction. Wrong direction means from where to where? Aorta to which ventricle? Left ventricle. Always be specific about that. Then you just draw out this inequality, you get your answer. You ask yourself, why is it pressure greater in here than the other one? Think about what is the thing that contracts. Okay? All right. Yeah, fairly straightforward. So over here, if you have ventricular systole, both ventricles are contracting. 1 and 2 need to be open, 3 and 4 closed. Simple as that. So it's answer is C. Yes. Okay. And shortest route. This one is actually linked to what we learned in primary school about human circulation. Our human circulation, they say elbow circulation. So it always has to return to the heart first. Leg to an arm. Leg, heart, lungs, heart, arm. This is A. Okay, very straightforward, Oh, beautiful. So if you can remember what we drew just now about the pressure differences resulting in the valves opening and closing, it will greatly help with this question. It will greatly help with this question. Okay, so Natalie, if I zoom in right onto this part over here, what valve open, what valve, what is valve that is involved over here? Is there a key here? Okay. Okay, I will tell you, this graph, right, is always going to be the same one. So literally, I just memorize, the highest one is the aorta, then the ventricle, then the atrium. That's it. The answer is there. Aorta, ventricle, atrium. You can either just remember this as like that, or you can also think to yourself, why is it like that? Why atrium lowest pressure? It's the smallest one. The walls are the thinnest. So the contractions are going to be least forceful. The ventricle are the largest muscles. So the contraction is going to have an immense increase in pressure. Okay? The aorta, more or less in a high range because it is constantly receiving blood from the left ventricle. So aorta, ventricle, atrium. How will this graph look like if it's about the right side? If I want to draw right atrium, right ventricle, and pulmonary artery, what would the graph look like? Yes, Jason. Very good. Right, ladies. So if I want to draw the same graph for right atrium, right ventricle, and pulmonary artery on this graph, the shape will be the same, but everything lower pressure. Okay, because the right side is the thinner muscles, so less forceful contractions. Okay, awesome. Keep that in mind. So question 2 here, we should be looking at aorta, left atrium, left ventricle, B. Okay, looking at B. Okay, let's go. Okay, which valves partner together? I have 4 valves in total. Which valves partner together? How do they partner up like that? 4 valves, how do they partner up? You sure? The 4 valves, how do they pair up? Yeah, so it's actually both AV valves. So bicuspid is on the left side, tricuspid is on the right side. They pair up. So if they will always close or open together, compared to the aortic and pulmonary valves. Those are also known as what? Semilunar valves. Okay, so here we must understand this one. Okay. So if semilunar valve opens, my bicuspid must close. They're opposite. Okay, if semilunar opens, bicuspid must close. Because why would the semilunar open? What will cause the semilunar valve to open? Contraction of the ventricles. So when both ventricles contract, semilunar must open to force blood out into the aorta and the pulmonary artery. Okay, when this occurs, I must ensure I don't have backflow to the atria, so I must close my bicuspid. Okay, right? So that's going to happen at the same time. We're just looking for the correct set of labels in the graph above. Jason, do you have an answer for this? Okay. 1, 4. Yes, very good. Okay, so valve close 1 and 3. So over here, okay, so over here we have bicuspid closing, okay, and then we have semilunar closing as well, okay, because the aortic pressure is now greater than ventricular pressure and do not want blood flow back from aorta to the ventricle. Okay. Okay, this one. Not what why not that. Okay, Ken. Let's go. Calm down, calm down, calm down. Tell your heart to relax. You know what's the average BPM? Around 60 okay, around 60 Sleeping can fall. Okay, maybe now to say now 100 scared. Okay, all right, come. Blood vessels A and B. Blood vessels A and B. A is the vena cava. Very good. B is the. I have 2 of these. They are the same. I have 2 B's. What are they? What are they? B is the pulmonary vein. Very good. Why do I have 2 of them? Why 2 pulmonary veins? Lini, why do I have 2 pulmonary veins? Pulmonary vein means what? connected to pulmonary. Pulmonary means what? Pulmonary. Lungs, lungs. So I have two pulmonary veins because one comes from each lung. Yes. Two lungs, what? Then the blood vessel must like that, ma. Yeah, so like that, lah. Then each lung then come back like that also, ma. Okay. Okay, awesome. Let's go. Function part level C. Okay, C just a valve backflow, blah blah. Let's move on. Okay, question 3. Here we are describing the entire circulation. So the entire sequence, yes. Describe and explain how blood entering the heart from the body organs reaches the lungs. What is our first point in describing the journey of blood? Okay, so it starts with vena cava and enters which part of the heart? Right atrium. Okay. So here I'm just gonna draw out with you, right? Because I'm gonna say vena cava to RA. After RA, go where? I'm just gonna draw with you. I'm just gonna draw with you first. After RA, go where? RB, fairly straightforward. Okay, RB. After RB, pulmonary. Okay, only the chambers I put letters. Everything else right. The whole thing, just two letter only. You know, Okay, pulmonary artery, go to the lungs already. Over here. Okay, so over lungs here, it's gonna get oxygenated. And how? What's next? After the lungs, I'm done. I have collected my oxygen. I have deposited my carbon dioxide. Very good. So now I have pulmonary vein into left atrium. Ventricle. Then okay, this is the entire chart. But the question says, very good. Reaches the lungs. So we only need to stop at here. So why does this question have 5 marks? What we have done so far is just describe the pathway. How do we explain? The blood pressure thing, the contraction things. Okay, so over here, maybe you can think of flows into a relaxed right atrium. All right, now relaxed right atrium. As the atrium fills up, the atrium then undergoes contraction. Then it will force blood into the relaxed ventricle, which subsequently contracts, forcing blood through the pulmonary artery into the lungs. Right, so this is how we can add on the explanation portion. Contraction, relaxation, pressure changes. Can? Can, All right, wonderful, ladies. Let's go. Want to look at B? The part B doesn't involve the diagram, so just jump straight down to it. Compare pressure of blood in the circulation to the body tissues versus circulation to the lungs. Firstly, what is the name of these two circuits? Systemic versus the word for lungs is just now pulmonary. Pulmonary circulation, systemic circulation. You can also refer to it as pulmonary circuit, systemic circuit. Okay. How do their pressures differ? Who has higher? Sorry? Systemic pressure is higher. Fact. Very good. Why? Sorry?
One is transfer extravascular.
Okay.
The other one is a lot more transfer.
Okay. Very good. Okay. So higher blood pressure to the systemic circuit. Importance versus why there is. The answer that I just gave is the importance of. The importance of having higher blood pressure in the systemic circuit, so it can bring blood, oxygenated blood to the body cells faster. And then while lower in pulmonary, so there's more time for oxygen to diffuse in the bloodstream. So that's the importance of versus why is there? Why is there higher blood pressure in the systemic circuit? Do you all see the difference in these two questions? Why is there a higher pressure in the systemic circuit? That's the important thing. Yes. Just sing. Yes. Okay, just sing. Come explain again. Why higher blood pressure in the systemic? Because good. Right, stronger muscular left ventricular wall results in more forceful contraction. That's why higher BP. Do you see the difference, right? Okay, thank you, Allegra. Right, so again, please keep in mind, learn to see what the language is in the question. Are we looking for why is there such a difference versus why is it important to have this difference? Can? Okay. Awesome. So yeah, partly is the structure thing. Okay. So pulmonary, systemic, portal. You only have no, this one digestive system one. Can you tell me what's the portal vein? The only portal vein you learn? Hepatic. That one is from where to where? That one is from liver. From small intestine to liver. Okay, hepatic portal is from small intestine to liver. This is the only vein in the body that doesn't go directly back to the heart. It goes to another organ first. Okay, no matter, not important over here. Let's go. Another pressure one. Okay, if you look at part 2, if you look at part 2 over here, again, if you can remember just now the pressure inequality thing. Remember just now the MCQ, the pressure inequality thing. Draft it out somewhere, use a pencil, write it on your paper elsewhere, right? Then you use that, answer your question here. So use that diagram, use that pressure change, the inequality to help you craft out your answer over there. Okay? Part 3. Why is ventricular pressure in the right lower than on the left? All right, now this one is the importance of, this one is the why. This one question say why. You see that? Question says explain why there's a difference. Okay, so why is there such a difference? Oh, right ventricular wall thinner, so less forceful contraction, lower pressure. Okay, so think about that. If the question says, oh, importance of, significance of, benefit of, advantage, then you talk about the I need to bring a greater distance, I need to bring it there faster, all these kind of things. Understand the language, right? Answering questions is not just about knowing the concepts, it's about learning the language of the subject as well. Okay. Okay. What is wrong with this heart? Describe to me what is wrong with this heart, please. The wall is too thick. So what? Very muscular. Wow. How we need to find out, we need to write stuff that we can use to say, oh, that's why bad. So wall is too thick, but I want to focus on something else other than the walls. Okay, so if the heart is this shape, okay, the normal heart this shape, the damaged heart also this shape. But if the damaged heart, the wall is thicker, overall shape is still the same, means that the space inside is the one that I want to focus on. Correct now. So if we start off with thicker walls, yes, then we must be able to veer towards not enough space. Why is that a bad thing? Ventricular space is reduced. What does it mean? Very good. Less blood can fill up the heart. So meaning, instead of, for example, if I say that, if in this amount of space, right, I can do this, I can move this amount of air, versus now I'm only allowed to do this. to be able to move the same amount of air, I must do a lot more times, right? So the damaged heart must now pump harder and faster. Okay, so usually at this point, right, it's not just like, wow, bigger muscle better. It comes to a point in time where the bigger muscle impedes its own movement, right? It will not be able to contract as effectively anymore. Okay, so here you want to focus on volume, right? The volume of blood that this heart can pump out per contraction is reduced. Okay, this person is likely not going to be able to engage in any vigorous activities. Okay, and this one not just say sit down there, wow, watch computer games and watch video, all can already. You cannot even have extreme emotions. You too happy, how? Heart pump faster or die. If too scared, how? Heart pump faster, also die. Right? So it may look like, yeah, okay, no need exercise, never mind. It's not a good thing, It's not a good thing, Okay. Okay, similar to that, but less disastrous. Hole in the heart. Anybody knows anybody that has a hole in the heart? Can I tell you right now? Most of us were born with a hole in the heart. And why now don't have? It touches up as we develop within the first few months. So only if there's a failure for the cell division over there, then the hole remains. Okay, right? So have you encountered this error damage before in your own notes and stuff? Okay, then I show you a bit. So over here, if you look at the diagram over here, what is mixing? Very good. Oxy and deoxy from left and right respectively. So the right side one doesn't matter because right side pump to where? The lung. And just taking oxygen, it doesn't matter what. It's the left side that is affected. Because now if there's a mixture, there is less oxygen that is in the left side. So my body cells receive less oxygen. This person also advised against vigorous activities. Okay? All right. Do you all know how to explain this? What are the two heart sounds? Lap dap. It's due to what? Is it due to the contraction of the muscle? It's due to the valve closure. Literally, the valve. This is the lap dap sounds that I heard. Okay, so it is the contraction of the ventricle slapping the AV valves shut. Then later on, when the ventricles relax, the two arteries want to backflow, cannot snap shut again. Okay, right? So it's a snapping shut of the valves that result in the heart sounds. Right. So over here, this one is about systole and diastole. So before I move on to respiration in the last 2 minutes, okay, my hand here, the heart alternates, not left right. The heart alternates up down. What do I mean over here? So when you have the heart contraction, it's not like that. If it's like that, it means you will always feel that this way. You will always feel that the body will be like rotate left, right. No, it's up down. So what happens over here? My right hand, the no watch one is atria, watch one is ventricle. What happens here? We have atrial systole, ventricular systole, both diastole. So if you do quick, right, tap tap. Okay, so this one forces blood into the ventricles. This one forces blood into the aorta and the pulmonary artery, ensuring, and at this point in time, it slowly starts to relax so that blood can constantly fill it up. Then both systole, both diastole. Okay, so it's like that, like that. Okay, it's not left side, left, right. The body will always be jumping around like that. Okay, so part 2 over here is asking how to describe that. So you talk about both atria contracting, ventricle relaxing, ventricle contract, forcing blood out, and you insert the lap dap respectively. Can? Okay. Pressure again. So I put a lot of pressure graphs in this handout because it is the one that is likely to come out also. It is the one that is the more complex one. Right? So it is likely for us to look at this. Based on this graph, what is the length of one heartbeat? How long is one heartbeat? Point 6. Okay, average is 0.6 to 0.8 per heartbeat. Okay, average is 0.6 to 0.8 per heartbeat. Okay, so this is something that we need to be able to identify as well. Okay, identify it as well. Okay, do you have time already? Blood vessel escape, blood vessel escape. Blood vessel also skip. Okay. Okay. Oh my goodness. Nobody after us. Let me just give me like maybe like extra like 10 minutes or so, okay? There you go. Later we have respiration. Awesome. First? Awesome. Then notice paper. Notice paper. Everybody have respiration? Are you on here? Okay, come. Go. And here you go. You only right? Yeah. Okay. Respiration. Respiration. Okay. Okay. Actually, you know what? Then we just discuss respiration. I didn't have time to get there. Okay, come on. What are some of the what? Oh, sorry, you don't have no. Okay, just take out there. Okay, what are some of the what do we learn in respiration? What's the you think back to when you first did this topic in school? Mehan? Yes. What comes to mind when you think of respiration? Versus. Okay, we actually learned 3 kinds of respiration. Okay, got aerobic, anaerobic. Why got 3? Aerobic, anaerobic is 2 only, right? Why got a third type of respiration? Yes. All right, anaerobic in yeast. Okay, so we have aerobic in humans, anaerobic in humans, anaerobic in yeast. Okay, right. So these are 3 types of respiration. So keep that in mind. What else can I think of? Let's go. Okay. What's the difference between breathing and respiration? What's the difference between breathing and respiration? Physical versus chemical changes. Right? Okay. Breathing, otherwise known as ventilation. You're bringing air in, pushing air out. Okay. So one idea that I need you all to try to appreciate more is that when I'm actually breathing, I'm not sucking air in. When I'm exhaling, I'm not pushing air out. So what am I doing then? What's the body doing then? In breathing. Okay. Must understand that air is a very noble character. We cannot force it to do anything. We can only invite it. So when I breathe in, I'm creating an environment in my lungs to invite the air in. So I create a high or low pressure in the lungs when I'm breathing in. All right, so when I'm breathing in, air wants to move from high to low pressure. I create low pressure to invite air in. And then the converse is true as well. So keep that in mind. We're always playing with pressure. So I'm not sucking air in. I'm just making a condition so the air will want to come in. Okay, and vice versa. Okay. What else can I think of? Okay, this one, carbonic anhydrase. Carbonic anhydrase is an enzyme that is found in the red blood cell itself. Very commonly misunderstood. Carbonic anhydrase is found in the red blood cell itself, so we must keep that in mind. Okay. One final big thing, maybe I can think of is the lactic acid thing. When is lactic acid produced or processed? Anaerobic in humans, must state because you got the yeast one, right? Okay. So what is the word equation? Tell me for anaerobic in humans. Word, not chemical word. No oxygen anymore. Anaerobic. So glucose, I give me lactic acid and small amounts of energy. Any carbon dioxide? Don't have. Keep that in mind. Anaerobic, no carbon dioxide only already. Okay. So when lactic acid is produced, what do I incur as well? What does the body incur? This concept. You know, the oxygen debt. The oxygen debt. When do I start clearing the oxygen debt? Only when you stop exercising. So breathing rate increases when you're exercising. The moment you stop, after you run, you do your last step already. You don't start it immediately. You don't do that, right? You're still maintaining panting like that. Your energy requirement doesn't remain high. It immediately drops. So why is the oxygen requirement still high? Repay the oxygen debt. All right, so keep that in mind. Right, so where does lactic acid go to get broken down? The liver. What do I break it down into? There's one option, glucose. Another option is carbon dioxide and water. Very good. So these are 2 options, but both options is better than lactic acid accumulation. So can I just confirm one last thing first? Your school, do they teach muscle ache and cramp? Is it due to lactic acid? Nothing to do lactic acid. Okay, then stick to it because actually it's not already. We found out it's actually due to the mineral deficiency. That's why you must drink all your histo everything, drink water, everything to balance out the minerals. Okay, just stick to your school syllabus first. Just say it's a lactic acid build up, then muscle fatigue and cramps. Can? Okay. Don't speed, don't speed. Solo. The water. Okay, very good. Okay, so that's the time we have for today for this. I'm sure all of us agree that it's very fairly cramped up, but that's the kind of how you can do over here. Go back and try the questions on your own time. I will send the answer key maybe in the next few days. So try out first if you want to. I will send it to you all. Wait, your exam is tomorrow. Okay, I'll send it to you all by today then. Okay, so go through and look through the questions. I'll send the answer key later today so you all can revise. Okay, so again after today, go back and discuss amongst yourself and see whether we can come together to form a class and learn together. Okay, but if not, thanks for spending your Labor Day with me over here. If not, that's all. Yep, friends at home. But if you heard what I said, yep, bye. Okay, so just text me if you need any help. Okay, bye bye. Bye everyone. Bye. Bye.